What's going on, everyone? My name is Jake, and in today's video, I'm going to be discussing the importance of osmosis and tonicity to ultimately help you guys understand these concepts for your first exam. So, before I get started into today's video, I want to go ahead and give a huge shout out to Chad's Prep, Ninja Nerd Science, and the Organic Chemistry Tutor for helping me understand what I know today. And if you guys need any help in your physical science courses, whether that's in introductory biology or an advanced biochemistry, I would highly suggest checking out their channels. I will provide a link in the video description to each of their channels in case you guys need some additional help. So without further ado, let's get into osmosis and tonicity. Alrighty, everyone. So in order to understand osmosis and tonicity, we have to first start by defining them. And so osmosis is the diffusion of water through a selectively permeable membrane where water moves from areas of high concentration to areas of low concentration. So we see here, we have water molecules over here in an extracellular matrix, and we have a phospholipid bilayer right here, which is a common selectively permeable membrane, where this water concentration is going to attempt to diffuse over the phospholipid bilayer and into the intracellular matrix, or the inside of the cell. And so notice how we have a high concentration of water over here on the left, and it wants to move to areas where there's less solute concentration, okay? And so that leads us with our next topic of tonicity. So what is tonicity? Tonicity describes how a solution's solute concentration affects water movement into or out of cells, okay? And so we are going to discuss a isotonic solution, a hypertonic solution, and a hypotonic solution, and how water movement affects all three of these different scenarios. So on your exam, you're probably guaranteed to have a question like this, comparing and contrasting the three. And what we need to know, so the first case over here on the left is an isotonic solution. And what's happening here is we have an equal amount of solute concentration on the outside as opposed to the water concentration on the inside of the cell. And so what's going to happen here is there's going to be no net movement of water, and we are going to have an equal concentration on both the inside and outside of the cell. And that's why there is no net movement of water. And a common example of a isotonic solution is an IV. So if you guys have ever been, if you guys have ever had to go to the hospital and they had administered IV fluids to you, this is a perfect example of an isotonic solution to prevent cellular damage. And yeah, it's pretty interesting. This one's pretty easy to understand. It's not too confusing, but the next two are where it can get a little tricky to some. So this next example right here, we have a hypertonic solution. So if we see here, on the outside of our cell, we have a ton of salt solute concentration, whereas on the inside, we don't have that much water, okay? Our water concentration is minimal on the inside of the cell. And so what's going to happen is we're going to have the diffusion of water out of the cell. And so we can see here when that occurs, water will begin to leave and the cell will begin to shrink. And basically, solute concentration is significantly higher on the outside of the cell, which results in shrinking. So if you guys have ever gone to the beach and accidentally ingested a bunch of salt water, this is exactly what can happen to your cells, because there's such a high concentration of salt on the outside of your cell as opposed to water, where the water on the inside of your cell is going to want to attempt to go out and diffuse to where there is less water, right? Because there's more salt and therefore less water, causing the cell to shrink. And an example of this, like I said, is just ingesting salt water. And so finally, we are left with a hypotonic solution. And so over here, we have a hypotonic solution where it's pretty much the opposite of a hypertonic solution. And as we can see here on the inside of the cell, we have an incredibly high solute concentration. Whereas on the outside, we have a limited water concentration. And so these water molecules on the outside of the cell, they're going to want to flow into the cell. And this can be a problem, right? Because the cell will begin to swell. There's an outward pressure that is created, which ultimately results in the cell bursting or lysing. And so an example of this would be drinking fresh water to the point where you develop water poisoning. It takes a significant amount of water for this to occur, but it gets to a point where your cells will literally begin to burst from all the water on the inside of your cell, or all the solute concentration on the inside of your cell, where water will flow in, causing the cell to burst or rupture. And so thank you guys for listening today. This is just a brief overview of osmosis and tonicity, so that way you guys can ace your first introductory biology exam. I hope you guys had a informative time here listening to this video. And if you guys, like I said, if you need any additional help understanding these concepts, make sure to reach out in the comment section or click on the links that I've provided in the video description. Good luck on your exam, guys. Take care.